Hey guys, welcome back to MedVids Made Simple. In this video we're going to see about anatomy of muscles. Now let's begin. The word muscles is derived from the Latin word mus, which means mouse. Muscles basically resemble the structure of a mice. So the belly of the muscle resembles the body of the mouse, and the tendons resemble the tail of the mouse. Muscles are defined as contractile tissues, which helps in movement. They basically help us to do all the routine activities, such as walking, running, lifting weights, etc. There are three main types of muscles, such as skeletal muscle, smooth muscle, and cardiac muscle. Skeletal muscles are found in the muscles in upper limbs and lower limbs, and in the body. Smooth muscles are found in the viscera, such as stomach. Cardiac muscle is found only in heart, and it's very unique compared to skeletal muscle and smooth muscle. First, let's see about the skeletal muscles. The skeletal muscles are also known as striated or somatic or voluntary muscles. They're called as striated muscles because they're known to have something known as striations or stripes. They're known as somatic muscles because they are present in the body. Soma means body, so they are somatic muscle. They are voluntary muscles because they are under the voluntary control by humans, so they do not function on their own. We are going to control them, so they are voluntary muscles. The skeletal muscles basically have an origin and an insertion. Origin is the point from where the muscles arises, and insertion is the point where they get inserted into some other point. So origin mostly remains unchanged. The muscle part which is attached to origin remains unchanged mostly during contraction. The part which is inserted into the other end is mostly what's going to contract to exert the action. So there are two main parts of muscles. First one is the fleshy part, also known as the belly. The second one is the fibrous part, which is either tendon or aponeurosis. The skeletal muscles includes two main tissues. The first one is the contractile tissues, which are nothing but the muscle fibers, which helps in the contraction of the muscles. The second group of tissues are the supporting tissues, which includes endomysium, perimysium, and epimysium, which helps to keep the integrity of the muscle fibers and helps them maintain the structure. So here we can see the structure of muscle fibers. Okay, so starting from the right side, okay, so you can see the muscle belly there. The muscle belly is covered by structure known as epimysium, which is marked in the picture. The epimysium contains many fasciculus inside it. Okay, so each fasciculus will be covered by something which is known as perimysium, which is also shown in the picture in the bottom. Okay, so inside each fasciculus there are various muscle fibers, and each muscle fiber is surrounded by something known as endomysium. Yeah, so when you take out a muscle fiber outside, it is covered by something known as sarcolemma, and inside which the cytoplasm is known as sarcoplasm. Each muscle fiber contains various small structures which are known as myofibrils, which contains finally the main contractile proteins such as actin, myosin, which helps in contraction of the muscles. This is the structure of muscle fiber. Okay, this is just to revise it. The muscles are basically composed of various fasciculi, and each fascicles are composed of various muscle fibers, and each fibers are comprised of several myofibrils, and each myofibril contains various myofilaments such as actin, myosin. Here we're going to see an individual myofibril. In each myofibril you can see the following dark and light zones. Okay, in this picture you can see a zone which is marked as A band, and you can see another zone which is marked as I band. The red zone, which is marked as A band, is actually the dark zone. The I band is actually, the yellow colored thing is actually the light zone. How to remember this? The word dark has A in it, so it is A band. The word light has I in it, so it is I band. It's very easy to remember. So, in the middle of I band, you can see a thick black line. It is known as Z line. Between two Z line, the part of the myofibril is known as sarcomere. In the middle of the I band, there is a line which is known as M line. And as I've told, the center part is the dark band, which is the A band. It has a center part which is light, which is known as H band. So let's revise what's present from left to right in a sarcomere. First, there is a Z line, next to which is a part of I band. Then comes A band. Inside the A band, there is a further light zone which is H band. Next to A band, there is some part of I band, followed by which there is one more Z line. It's very easy to understand. If you don't get it, watch it once more and you'll get it. So here you can see the contractile proteins such as actin and myosin. The filaments which are marked in pink are myosins, and those which are marked in blue are actin. Okay, so the actin comprises of the light portion and the myosin comprises of the thick portion. So it's the thick filament. The actin is the thin filament and myosin is the thick filament. So here you can see the black crossed lines, black zigzag lines in the picture at the top. That line is the Z line which we've shown in the previous picture. So between two Z lines is something known as sarcomere, as I've told you earlier. So in this picture you can see that they are represented one sarcomere which has alternating actin and myosin. So you got to remember the fact that each myosin filament is surrounded by 6 actin filaments. Now how does muscle contraction occurs? Muscle contraction occurs by overlapping of the actin myosin filaments. So all these come close together and it will result in contraction of the muscles. The A bands are dark bands and I bands are light bands. I've told you earlier how to remember this. The dark word has a in it, so it is A band. The light word has I in it, so it is I band. The Z line is also known as Krauss membrane. You got to remember this point. So there are 3 main types of skeletal muscle fibers. First one is type 1 fibers, which are slow fibers. Second one is type 2 fibers, which are fast fibers. And type 3 is intermediate fibers. The intermediate fibers are nothing but a variant of type 2 fibers. First, let's see about type 1 fibers. The type 1 fibers are slow fibers. They're called so because they cause slow tonic contraction. Okay, they have the property to undergo slow tonic contraction. So they are present mainly in postural muscles. They're involved in maintaining the posture. They are red in color because they have a large amount of myoglobin. They're also rich in mitochondria and oxidative enzymes. So they basically produce their energy by oxidative mechanisms. They have very little phosphorylases and they are highly resistant to fatigue. So they won't get fatigued so easily. That's why the postural muscles will help to maintain the posture for a long time without getting fatigued so easily, and that's why they contract very slowly. The type 2 fibers are known as fast fibers because they undergo fast phasic contraction. They're mainly present in areas where large-scale movements are required. So they are paler in color. That's because they have less amount of myoglobin. They also have lesser number of mitochondria and oxidative enzymes compared to the type 1 fibers. They have more glycogen and phosphorylases. They easily get fatigued because they involve glycolytic pathways for the energy production as compared to type 1 fibers. The type 1 fibers uses oxidative pathways. So in this picture we can see the three types of muscles. The first picture shows cardiac muscle, the second one shows skeletal muscle, the third one shows smooth muscles. The skeletal muscles are striated muscles, as you can see in the second picture in the middle. They're long and cylindrical. Their fibers are unbranched. So compare it with the first picture, which is cardiac muscle. There you can see very well that the fibers are branched in cardiac muscle. Okay, so the skeletal muscles are also multinucleated. As you can see here, each fiber has many nucleus. Okay, and they also have light and dark zones, which we've talked about earlier. And the very important feature is striations, which are present in skeletal muscles, but these striations are not present in smooth muscles. Okay, now let's see about smooth muscles, which is given in the third picture below. So smooth muscles are spindle shaped, and they are mainly present in organs such as esophagus, the distal part, stomach, iris of the eye, etc. They are uninucleated as compared to skeletal muscles, which are multinucleated. They do not have alternative light and dark bands as compared to skeletal muscles. These are very important points regarding smooth muscles. Now let's see about the very important muscle, cardiac muscle, which is very unique. They are present in the myocardium, that is the wall of the heart. These muscle fibers are short and cylindrical, and you can see that they are branched. They are also uninucleated, such as smooth muscles. Okay, so they have very faint light and dark bands, and they have a very unique feature which are known as intercalated discs, which are present only in cardiac muscle. So if someone asks you where can you find intercalated discs, you can straight away say that they are present only in cardiac muscle. So these are the three main muscles, and we have talked about the differentiating features in these three types of muscles. Now let's see about the nerve supply of the various muscles. As you all know, skeletal muscles receive a major nerve supply from motor nerve fibers. They receive 60% nerve supply from motor fibers, and they receive 40% nerve supply from sensory fibers. The smooth muscles are classified into single unit type and multi-unit type based on their nerve supply. Okay, so they can be classified into single unit type and multi-unit type based on the nerve supply. The single unit type is basically seen in intestines. So in this type, the nerve supply is usually sparse or very little. Multi-unit type is seen in muscles such as those in ductus deferens. So each muscle cell in this system receives a separate nerve fiber, so the nerve supply is very rich in multi-unit type. So what's the main differentiating feature? In single unit type, the nerve supply is little, whereas the multi-unit type has very rich nerve supply, because each nerve fiber is separated by, each muscle cell is separated by a separate nerve fiber. So now talking about the interesting part, the cardiac muscle. The cardiac muscle's nerve supply is not under our control, so they are not controlled by our voluntary activities. It's basically involuntary, so it's controlled by the autonomic nervous system. It's predominantly by the parasympathetic nervous system and comparatively little amount by sympathetic nervous system. The parasympathetic nervous system helps to control the heart rate and keeps it low. Sympathetic nervous system tries to increase the heart rate. Okay, this is the nerve supply of all the three muscles. An interesting fact to know is that a heart which is denervated, which means when you cut the nerve supply from the heart and take it out of your body, the heart supply increases rapidly. This shows that the nerve supply of the heart is mainly from parasympathetic nervous system, which keeps the heart rate controlled. And when you cut the nerve supply, what happens is the inhibition is lost. The inhibition done by the parasympathetic nervous system is lost. So the heart rate will increase when you denervate, and which means you cut the nerve supply of the heart and take it out of the body. Please leave a like to this video if you liked it. We'd like to thank our contributor Bearers M for donating us $80. Help us make more videos by donating us through Patreon. Please visit patreon.com/MedVidsMadeSimple and find out why you need to donate us. And also check out various rewards you get by donating us on Patreon. Thanks for watching this video. Please subscribe to our YouTube channel MedVidsMadeSimple and share this video to your friends. And please ask your doubts in the comments below. Thank you.